of spring 2020, April 20th. We're still in our pandemic coronavirus shelter in place. Um, we are going to start this week our unit on prion diseases, and hopefully we'll also start Wednesday and Thursday our unit on medical microbiology. Most of the information on prion diseases, I think a lot of you already have a pretty good grasp on this, so hopefully a lot of this will be a review. We do have a few more details to throw in there. There's a study guide posted on our Canvas site, so if you want to be most efficient with studying for prions for our lecture exam 3, I would suggest you use the study guide. Okay, folks, so again, we'll just kind of walk through this one slide at a time. So we are discussing disease-causing prions, not the normal cellular prions. And prion was a term coined by Stanley Prusiner down at UC San Francisco, and it stands for proteinaceous infectious particles, or it's supposed to. We want to remember that prions are acellular. They lack nucleic acid. They're abnormally folded cellular proteins, and sometimes this is confusing for folks. So cellular here is an adjective, meaning that these are proteins that cells make, right? But they aren't cells themselves. They're found in high concentration in neurons, nervous tissue, and we can also find them in lymphoid tissue, and in some of the diseases we'll see we can even find them in white blood cells. In this simple cartoon, if this represents a good normal prion, we can see it's rich in alpha helices here. These broad blue arrows represent beta-pleated sheets, so we can see in the good normal cellular prions, rich in alpha helices, relatively low beta-pleated sheets, and the normal prions can be denatured, for example, by autoclaving. But when they misfold, we see a reduction in the amount of alpha helices, we see an increase in the amount of beta-pleated sheets. So if I were to ask you on an exam to compare the structure of the good normal cellular prions with the misfolded disease-causing prions, we want to remember that the primary structure, the specific amino acid sequence, is the same, but secondary level of structure has changed, tertiary level of structure has changed, and we'll see eventually we're going to add quaternary level of structure, right? So even though the amino acid sequence is the same, it's the abnormal folding that causes the neurological diseases. So the misfolded prions, they cause a family of diseases, fatal neuronal degeneration, called TSC for transmissible spongiform encephalopathies, right? And we want to remember one of the bad things about them is they're very, very hard to denature. They're very hard to inactivate. As we mentioned earlier, it was Stanley Prusiner at UC San Francisco who first proposed our current understanding of prions, right? He received the Nobel Prize for it. He was one of these folks that literally thought outside of the box. And again, folks are often amazed when we discover that we have a gene that makes the normal cellular prion protein. And you don't have to know this for the exam, but it's the PRNP gene. And it makes the good normal cellular prion. And they have abbreviations here. So the good normal cellular prion, they call it PRP superscript C for cellular. And then when it misfolds, they call it the disease-causing prion, they call it PRP superscript SC for scrapie. And scrapie was the first prion disease that was discovered. It's a prion disease of goats and sheep. I shouldn't say maybe discovered. It was the first prion disease that was described. So we would say that these, again, are protein misfolding diseases. This was just, I was trying to find an illustration of where prions are found. So here, folks, this is at a synapse. This is the presynaptic neuron. Here's a postsynaptic neuron. And we can see, guys, here's our little diagram here. That we've got prions here. We've got prions here on the neurons, prions there on the neurons. So they're going to be found in rich abundance on neurons, cells of the nervous system. So with regard to how the prions misfold, one hypothesis. One model is that a misfolded prion binds to a good normal prion and somehow causes the good normal prion to misfold, right? And then we have like this exponential or logarithmic conversion of good normal cellular prions into the misfolded disease-causing kind. And as we mentioned, folks, not only do we have changes in secondary and tertiary structure, but the abnormally folded prions can then stack. So they're achieving that quaternary level of structure that normal prions wouldn't have. The misfolded prions, they can stack, forming long filaments. Okay, so these are the little filaments or the fibrils. And the fibrils can cluster together in nervous tissue, creating this plaque. And somehow, I'm not sure how, and I'm not sure maybe if we know yet how these tangled masses of fibrils trigger the holes to form in the brain, causing the spongiform encephalopathy. Or it might be that there's another mechanism for the neuron cell death. Maybe because the prions can't carry out their functions, perhaps this triggers a neuron cell death. So what we can see is literally the holes that form in the brain. So these would be samples, thin sections through the brain that have been stained, and we're looking at them with a light microscope. So this is a normal brain. We can see we've got a lot of cellular material. There's no holes. So here's the brain of a human that's died of a prion disease called Kuru. And again, you guys, we can see these huge holes. Just devastation here. This is the so-called spontaneous classic form of Creutzfeldt-Jacobs disease that us old people can develop spontaneously. We're not sure why or how. And again, we can see these holes. It is interesting how the Kuru and the classic form differ. Kuru is an ingestive form, right? And then over here we have Scrapie, which is also an ingestive form of prion disease of goats and sheep. And it is, to me, it's kind of interesting, guys, is Kuru is an ingestive form of prion disease, as is Scrapie, and it looks to me like the pathology in the brain is similar here, and it's quite different to the classic form. So because the brain develops holes like a sponge, the TSCs are called spongiform. Something looks like a sponge with holes. Encephalo refers to the head, specifically the brain. Pathy is an abnormality, so so-called holy brain disease, and transmissible because unfortunately we now know it can be spread from an infected host to an uninfected host. So TSCs, transmissible spongiform encephalopathies, spreadable holy brain diseases. And unfortunately, they're 100% fatal. We don't have cure, we don't have a treatment yet. So again, folks, with regard to why the holes form in the brain, again, I don't think they truly understand. Again, it might be because the prions can't carry out their normal function, so there's an interruption in signaling events between neurons. But again, those abnormal prions, somehow they're triggering that cell suicide, programmed cell death, or apoptosis of the neurons. And this is really important, you guys. Do remember this for our lecture exam 3. Our immune systems do not recognize the misfolded prions as being foreign, right? Our immune systems still see the misfolded prions as self, and therefore there's no immune response. We don't have an inflammatory response. There's no antibody response. And this, not only is this bad because it's like we have no natural defense against the misfolded prions, but it also makes it really tough then to run diagnostic tests when a human or an animal is still alive. Many, as you might recall from our pandemic corona diagnostic tests, we can use antibodies as evidence that a person has had been infected, maybe still is infected with a pathogen. But with the misfolded prions, since there's no antibody response, we can't screen humans or animals to see if they are infected, right? Because they don't make any antibodies. So this makes it really challenging coming up with a diagnostic test, right? There's no quick and easy blood test, which ideally we would love to have. A little bit of history of TSCs. You might say, we might go back in history, guys. So with regard to records of prion diseases, the first records we think come out of Britain. as early as the 1700s, when farmers would describe a weird disease in their sheep that they would call scrapie. And the reason it was called scrapie is it appeared that the sheep maybe had like an incurable itch. You know, they would scrape themselves against walls or trees or the sides of a barn, scrape their wool off and start bleeding. Same disease in goats. Um, or it might be that because the sheep and goats, the destruction in the cerebellum, they're off balance. And so they're actually scraping themselves along, you know, fences or barns, trying to stay upright. That might be another reason for that. And then in the 1920s in Europe, Creutzfeldt and Jakob described a fatal neurological disease. Didn't know the cause, right? Didn't know the cause of scrapie. Didn't know the cause of what became known as Creutzfeldt-Jacobs disease, CJD, right? And in the 1950s, we're going to jump to Papua New Guinea, where a horrible disease of the Fore people were discovered called kuru for laughing or trembling, right? And nobody knew the cause of that either. And um, we'll see that the link is all three of these diseases were caused by prions. So with kuru, again, it was identified in the Fore people of Papua New Guinea, and it was finally determined that how kuru is being transmitted between the people of the tribe was through a practice called mortuary cannibalism. And this is where when a loved one, a family or member of the tribe died, the women would prepare the body. It would be cooked, and then the body would be consumed. And it's believed that because the women prepared the bodies, you know, with knives, they would be cutting themselves. And also my understanding is that the women and children would eat the brain and the spinal cord, which would be the parts of the body richest in prions. And then the men, if the men consumed the body, they were more likely to eat skeletal muscle, meat, which would still have prions, but would have lower concentrations. So as a result, the women and the children were devastated by kuru. In the documentary, there's a link here in another slide. One of the elders said, you know, one time there was like no women around. You know, they'd all died of kuru. So one hypothesis is kuru may have started from one of the older members of the Fore people dying of the sporadic form of prion disease, the sporadic Creutzfeldt-Jacobs disease. And again, this is when, as we age, our prions misfold spontaneously. Again, we're not exactly sure why, but because the tribe practiced mortuary cannibalism, when the person that died of the sporadic CJD, when tissues from his body was consumed, then that started this new prion disease transferred through ingestion, right, through eating the meat, the tissues that had the misfolded prions. And then those misfolded prions would bind to the good normal cellular prions of the new victims, right, and cause them to misfold. And then we developed this new ingestive prion disease, which we call kuru. So kuru for trembling. And then when those poor people died, right, their bodies would be consumed, spreading the misfolded prions to more and more members of the tribe. So this was just horrible. Yeah. Our cat is here listening. Okay, and folks, this is if you have time, this is a really powerful documentary, an Australian documentary, um, documenting the history, the research that went into um, proving that it was mortuary cannibalism that caused kuru, and some of the crucial work. It has it's really powerful and truly heartbreaking. They show members of the Fore people suffering with kuru. It's a long documentary, but it has, I think, a lot to be said. They talk about with the bovine spongiform encephalopathy, mad cow disease, and now we know that those BSE prions can be transferred to humans. It goes into kind of like the future of what's going to happen with prion diseases amongst humans and animals. So if you can, it's an excellent documentary. It is definitely a heartbreaker. So if you're feeling kind of down, maybe don't watch it yet. You know, wait for some time when maybe things aren't so tough here at home. Okay, so then folks, just focusing then on, we've talked about Kuru, right? So we know that that's transferred through mortuary cannibalism. So then let's look at some other human prion diseases, and we'll focus on the Creutzfeldt-Jacobs disease. So there's about 1 case per million people, and we could break them down roughly into 4 types. So there is one form, this is really tough one, you guys, inherited or familial, and this is an autosomal dominant gene. So that means if you inherit one of the genes that will encode for a prion protein that will definitely misfold, all you need is one copy of that, right? So of the 1 case per million, 10 to 15% are considered to be this inherited or familial form. And then iatrogenic, I guess they don't use this term much anymore, but it literally means caused by the doctor. So iatrogenic would be when you go in for a medical procedure, or maybe you receive some kind of transplant, and it's the medical procedure, maybe through contaminated surgical instruments or through the transplanted tissue, that you become infected. And again, of the 1 case of a million, they think about less than 1% of those cases are iatrogenic, again, caused by some medical procedure or treatment. In the old days when I was growing up, if kids weren't growing well, they would take pituitary extracts from people who had died, which would contain human growth hormone, and they would inject that into kids to try to get them to grow. And sadly, that was one way that prions could be transmitted from a person that had died of Creutzfeldt-Jacobs disease, right? The misfolded prions are in the pituitary extract and injected into kids. And then organ and tissue transplants, so dura mater transplant, that tough covering of the central nervous system, the brain and spinal cord. Again, these would be um, people, when people died, they could donate their dura mater for maybe accident victims, or maybe folks that had damage maybe through cancer. And then corneal transplants, these are all tissues that contain the misfolded prion. So if you receive these contaminated tissues, then you could develop the iatrogenic Creutzfeldt-Jacobs disease. And then CNS electrodes, these can't be properly decontaminated. So if they're reused again, they're on the brain of a person with a prion disease, then they become contaminated, and then they can transfer the misfolded prions to the next patient. Most of the Creutzfeldt-Jacobs disease are the so-called spontaneous or sporadic form, and this represents about 85% of the cases. And as far as we know, it's this spontaneous misfolding as we get old. So with regard to genetic risks, it's a little gray still. It appears that in the amino acid sequence for your normal cellular prion, if you have a methionine, it amino acid position 129, that might increase your risk for misfolding, but a lot of people have this amino acid methionine at position 129, and not all of us develop the misfolding prion diseases. But what's really important, you guys, is the sporadic form. It's supposed to occur only in us old folks. So older folks, like maybe starting around 50 years of age, right? So this is not a prion disease of young people, right? This is an old people's prion disease. And the reason that that was important was in the 1990s, when in Great Britain, young people, like teenagers, early 20s, started presenting with what looked like Creutzfeldt-Jacobs disease. The doctors knew something was really wrong. This couldn't be the sporadic form. And eventually, this form became known as variant Creutzfeldt-Jacobs disease. And it was eventually discovered it was an ingestive form of prion disease. Young people had become infected through eating beef that contained the mad cow or bovine spongiform encephalopathy prions. And the heartbreak here is for many years, people were saying, no, you cannot get a prion disease from eating beef from a BSE infected um, cow or bull or steer, and that was just wrong. I just threw this in, folks. You don't need to know these details, but just a way to compare how the prions are acquired. So again, this is the ingestive form, the variant Creutzfeldt-Jacobs disease. right? This is the iatrogenic form, right, from contaminated instruments or um, contaminated tissues or organs. Over here, again, you guys, we can see that with the variant Creutzfeldt-Jacobs disease, the average age is around 27 years, whereas with the sporadic form, in this old folks, it's around 65. And this just gives you an idea of some of the initial signs and symptoms. So it's mostly related to abnormalities in the nervous system. And this, to me, you guys, you know, I I obviously don't know my anatomy very well, but to me this is fascinating that the different human prion diseases should affect different parts of the brain, right? I just find that so fascinating. And again, folks, okay, so cerebrum, right, and cerebellum, to me it's really interesting that the two ingestive forms of human prion diseases, so Kuru and the variant Creutzfeldt-Jacobs disease, both of them target the cerebellum, right? And it looks like the variant Creutzfeldt-Jacobs disease also goes after the cerebrum. But I just find that fascinating. It's like, why? Why do the different human prion diseases, why do those prions affect different parts of the brain? This is a little disturbing, and granted, you guys, it's like we don't, well, let's see here. No, 16, 17, 18. So 2019. This is something that we do need to remember is that prions can have really, really long incubation periods. So this is talking about um, the sporadic form. So the number of people dying as a result of sporadic CJD in the UK is increasing every year. So one concern might be, are some of these what are being described as sporadic CJDs, are they actually variant Creutzfeldt-Jacobs disease that have really long incubation periods? I know that if you go to donate blood, they'll ask if you have lived in Great Britain during a certain interval of time when the BSE, the mad cow, was going on. And if you lived there during that time, they will refuse to take your blood. And the fear is that if you ate contaminated beef during that time in Britain, you might have gotten infected with a disease-causing prion, and it might be that it just has this really long incubation period. So they don't want to risk taking your blood and then transferring it into a recipient, because we know now that you can, especially in these ingestive forms, you can find the misfolded prions on white blood cells. So again, people are interested if there's genetic predisposition, excuse me. So we said that it does seem like that if you have this methionine at the amino acid position 129 in your normal cellular prion, it might increase your risk for misfolding, right? But almost half of us have that amino acid sequence, right? And not half of us, or 40% of us, do we go on to develop Creutzfeldt-Jacobs disease. So there must be some other trigger, and we just haven't figured it out. So again, we could say problems with the misfolded prions. They're really resistant to destruction. They're very hard to denature. So people always ask, well, if I eat beef, and it has a BSE mad cow prion in it, what if I cook it really well? Cooking won't destroy them. Heating won't destroy them. Boiling, rather, I should say, won't destroy them. And the prions, again, they're going to be present in these ingestive forms, not only in the nervous system, so brain and spinal cord, but you're also going to find them in the muscle, the meat, and lymphoid tissues. And I think later we have a slide showing you which parts of cattle you shouldn't eat, because they're the parts that are going to have the greatest concentration of prions. So ultraviolet won't destroy them. They run experiments with the prions, right? So prions just laugh at UV. Normal autoclaving. This is something to remember, you guys, that normal autoclaving, and normal autoclaving is 121 degrees Celsius, 15 pounds per square inch of pressure over atmospheric pressure for 15 to 20 minutes. That will not inactivate the prions, and that's really kind of nightmarish for hospitals. So again, we can have the iatrogenic form of Creutzfeldt-Jacobs, where there's transmissions in hospitals by contaminated instruments, contaminated tissues, and organs. And also we know that the misfolded prions can be found in blood in the ingestive forms. So, people are always so interested, well, what can inactivate the disease-causing prion? So these, you guys don't have to remember this, but just to kind of give you an idea. So there's protocols here where if you soak your instruments ahead of time in sodium hydroxide or sodium hypochlorite, bleach basically, you soak them ahead of time and then autoclave, right, and clean and rinse, and then autoclave again, that they found that this will inactivate the disease-causing prions. But the problem is a lot of surgical medical instruments, they're so delicate they can't withstand this, right? So there's some medical instruments that would be destroyed by this treatment. So there's a lot of research going on to try to find other ways to decontaminate medical instruments that might be contaminated with prions. So for sure, there's incineration. In fact, when I first started teaching, when we were talking about prions, one of the students who worked on a surgical unit said that if they do any kind of surgery on a patient with a known prion disease, they just incinerate everything. They don't try to clean and autoclave the instruments. Everything gets incinerated. And then my understanding is in the European Union, they have developed special enzymes that can help remove the harmful prions from medical instruments, right? But as far as I know, those have not been approved for use here in the United States. So with regard to diagnostics, remember the bad news is there's no immune response, there's no antibodies. So we can't just take a blood sample and look for antibodies to the prions, that's just, we need a blood test. Right? So, we might start with behavior changes, right? Before the person or the animal is dead. And then after the person or animal is dead, you do the brain histopathology, right? But here again, the victim would be dead. You can, when animals are alive, right? Antemortem is when they're still alive. In cattle, to screen cattle, you can do tonsil biopsies. Probably not the easiest thing to do, but at least you can screen before the cattle are killed, looking for the ingestiform of the mad cow disease, BSE, and then taking samples of lymphoid tissue. Again, this would be while the animal's alive. So the so-called third eyelid in sheep and goats, and also rectal biopsies. You can look for in this case, this would be the scrapie prion in sheep and goats. So this, again, folks, you don't need to memorize this, but just so much work going on and trying to develop new tests to detect prions. The earlier we can detect the diseases, the better, right? So this was just a skin test in rodents, right? And then with another thing we need, of course, would be treatment. We don't really have any successful treatment yet. So there have been some experiments using quinacrine, an anti-malarial drug, and chlorpromazine, which is an antipsychotic drug. So in animal models, it seems to slow down the progress of the prion disease. But I don't think either of these drugs is shown to be beneficial to treat prion diseases of humans. And again, folks, this is trying to look at antibodies, antibody production. Can we develop a vaccine against the prions? That would be awesome, right? So just lots and lots of work going on here with regard to the prion diseases. And one reason you might say, well, prion disease is not that common, one case in every million people. But another reason we're so interested in the prion diseases is that there's several other, maybe much more widespread diseases. For example, Alzheimer's disease, so widespread. Parkinson's disease, amyotrophic lateral sclerosis, Lou Gehrig's disease. These diseases are also protein misfolding diseases. And indeed, sometimes you'll hear people talk about Alzheimer's as another type of prion disease. So we want to remember, you guys, that the protein involved in Alzheimer's disease, the misfolded protein found in Alzheimer's disease, it's encoded by a different gene, different from the prion protein, but just like prion diseases, this is a protein misfolding disease, a proteopathy, right? So the prion diseases that we've talked about here, we could say are similar to Alzheimer's, Parkinson, and amyotrophic lateral sclerosis in that they're all protein misfolding diseases. And maybe if we found a breakthrough, like how to prevent prion diseases or how to treat prion diseases, maybe that would be a breakthrough so we could learn how to treat or prevent Alzheimer's, Parkinson's, and Lou Gehrig's disease. That would be awesome. So just going, we've just gone over the human prion diseases, you guys, so now we'll just go over some of the animal prion diseases. So prions can be zoonotic. We want to remember that the mad cow, the BSE prion, can be transferred to humans, and so the BSE prion is zoonotic. The oldest prion disease of sheep and goats, so the scrapie or scrapie prion, as far as we know, it's not zoonotic, but it's like, you guys, I would never trust a prion, right? Anyway, so we'll just go back here. So folks, you'll remember that we said the reason this prion disease was called scrapie is it appeared that the sheep or goats would scrape against, a rock, a tree, a fence, a barn until they scraped their wool and their skin off. They could start bleeding. And of course, this is bad because in the blood, the scrapie prions would be located, and thus the environment gets contaminated with the prions, right? And especially some rough surface that another animal might come along and rub against, right? So in the Kuru video, I think they show you some scrapie infected sheep, and in some ways they look like the Kuru victims because you could tell they're off balance, they're falling over, right? So that might be one reason they're scraping themselves against the walls to stay up. But also, indeed, some people think they really are pruritic, so meaning they have this incurable itch. They'll have tremors. Incubation period 2 to 5 years. So here we see this long incubation period again. And once the clinical signs develop, usually the animal dies within about 6 months. So, of huge concern is the placenta, and the placental fluids are chock full of disease-causing prions. So when the mama ewe or the mama doe give birth, herd mates will come over and investigate, and they'll investigate maybe by chewing on the placenta, right? And certainly the grass where they graze is going to get contaminated with the prions, right? So it's again through ingestion that we think that the scrapie prions are transferred from one member of the herd or the flock to the other. And again, huge concerns, you guys, with the environment becoming contaminated. And those prions are so resistant, they may remain infectious for years and years and years. We want to remember that milk and colostrum is the first milk that mammals make. It's chock full of antibodies. Milk and colostrum also contains white blood cells, right? So unfortunately, the milk and colostrum are also prion positive. And then we'll show folks that a common practice is dead animals, if they can't be sold for food, dead animals, their carcasses are boiled up to make a protein-rich, what's called bone and blood meal. And this bone and blood meal, it's used in gardening. And we'll see it's also used as a protein supplement in animal feed. So that's another way that the prions can be passed from one animal to the other, is through this contaminated bone and blood meal. This was just to give you an idea, folks, that historically we have had scrapie here in the United States. There's been an eradication campaign, which means that if any flock is identified, all the animals are killed. This was, let's see here, obviously this is almost like 10 years old, right? No, more than 10 years old, 15 years old. But this was the distribution of scrapie positive herds and flocks in the United States, right? So we have had scrapie here in the United States. In the report I saw of 2018, there was only one flock that was still scrapie positive in Pennsylvania, right? So they are making progress there. So with regard to after the animal's dead, they look at gray matter of the brainstem, and in live animals they'll look at lymphoid tissue, either a rectal biopsy or a third eyelid biopsy. And again, once a flock is identified, all the animals will be killed or quarantined. And again, you guys, they've just made a lot of progress in trying to eliminate scrapie from the United States. This is kind of a tragic story. This is the BSE stands for Bovine Spongiform Encephalopathy. Bovine refers to cattle, right? So this is the so-called mad cow disease, and this is kind of my twisted sense of humor. Here are two misfolded prions, and they're saying we could never have done this without the humans. So these would be two little BSE prions, right? And we're going to see what did we humans have to do with the emergence of BSE. So this slide, you guys, usually if we're in lecture, I read this because I think this is such important information. So if you'll just bear with me here. So this is called the Great Recycling. From horns to tails, industries recuperate everything. Many products find their way back into the food chain, human and animals. Safe? Not so sure. Well, we know for sure, you guys, it is not safe, right? Even for cosmetics or medical products, the threat from prions cannot be disregarded. So we'll just take this kind of from the start, you guys. So this would be our farms, right, or our feed yards. So cattle, sheep, and goats represent ruminants, right? They have those four compartmentalized stomachs. Pigs and poultry aren't ruminants, right? So what will happen is if the animals are healthy, right, they would be sent to slaughter, right, and then their meat would be sent to the stores where we would buy it, right? But then there's some parts of the animals that are considered inedible, or if an animal is sick or dies on the farm, you can't use um, the meat for human consumption. So they have a process called salvage, and in Britain, the folks that carry this collection out are called the knackers. So the knackers process non-edible animal remains, bones, skin, blood, feathers, rind, offal, as well as bodies of animals collected from farms. So these would be animals that would die on the farm. The waste is mixed and cooked using methods which are not always guaranteed to be effective against prions. So they aren't, they aren't. They are not effective against prions. So if this is boiled up, remember prions are resistant to boiling, right? So if the cattle have BSE, or if the sheep or goats have scrapie, right? All of that is going to go into this animal byproduct. Now let's see where the animal byproduct goes. And this is what's mind blowing, you guys. So millions of tons of animal waste are recycled every year is what this big pot is telling us. So where did these animal products go? So various extracts. Elastin, collagen, beauty creams, glycerin and toothpaste, lipsticks, some soft drinks, keratin and shampoos, tallows, animal fats, omnipresent, means they're everywhere in foodstuff. Biscuits, which I think the Brits called cookies, biscuits, ice cream, stock like soup, cooking oil, also used for making soap. Gelatin, from sweets to pâtés, desserts to medicines, even for preparation of table wines, right? So Jello gelatin, that's an animal protein that we love to eat as kids. Jello gelatin. Animal feed. This is where we really bad news. So millions of tons are produced each year, prohibited for cattle and sheep, reserved for pigs and poultry, and also fish, for which it is presumed there is no risk. We hope, right? So this would be that blood and bone meal, that high protein supplement that would be added to animal feeds, right? Is a cheap source of protein, and we see this is the connection then between scrapie and BSE, or mad cow disease. So this was the best picture I could get, you guys, and I think this might be German, right? So don't think you're going crazy. This is not in English, but this is just a diagram showing how when sheep or goats were infected with scrapie or died of scrapie on the farm, their bodies then were made into those recycled animal products, and some of their bodies then would be made into that bone and blood meal, which would be added to cattle feed. So then the cattle would eat it, and what we now think happened was the scrapie prions that weren't inactivated by the cooking. Now the scrapie prions are in the cattle feed. We fed the scrapie prions to the cattle, and the scrapie prions then were able to bind to the cattle's prions and cause them to misfold. So then we have this brand new disease of cattle, bovine spongiform encephalopathy. Now, you guys, ruminants are herbivores. They're in nature, all they would do would be to eat plants. So cattle wouldn't be normally eating protein made from the bodies of sheep or goats. So this was where we humans got involved, where we kind of messed things up here, right? By feeding the blood and bone meal made from the scrapey sheep and goats to the cattle. So now the cattle have their own bovine spongiform encephalopathy, prion, the so-called mad cow disease, right? So the mad cow disease, it appeared in the 80s in Britain, right? And it was just heartbreaking because the British agricultural minister said, hey, you don't have to worry, you can still eat British beef because the mad cow disease can't jump to humans. And now we know that was totally wrong. So what happened, it was within 10 years, then this new prion disease of young people emerged. So that they call it new variant. Now we just call it variant Creutzfeldt-Jacobs disease. So these young teenagers, people in their 20s, start presenting with Creutzfeldt-Jacobs disease, and the doctors knew something was horribly wrong. Right? They're too young to develop the sporadic form. So eventually, it was finally determined that it was indeed that these young people had become infected from eating beef from cattle that did have the BSE prions, right? And here, you guys, these are just like high risk tissues from cattle. I would say from almost any animal. Be careful of. So the brain is going to have a lot of prions. The spinal cord is going to have a lot of prions. Lymphoid tissues, say of the intestines and also the tonsil. So beef tongue, you guys, beef tongue is kind of a delicatessen food, so I would worry when they cut out the tongue, are they going to get some tonsil in there? They could have prions. If you think of hot dogs or sausages, folks, historically, the casings are made from intestine, right? So I was like, I don't know, that's such a great idea. And then, folks, I know this is really disturbing, but in slaughter yards, one way when they have cattle coming through, they have bolt guns that they use when they pull the trigger. A bolt is driven into the brain of the animal, and that's what kills them. But they've shown that the bolt guns, that bolt drives into the brain with so much pressure, it actually drives um, tissue from the brain into surrounding tissues, like even all the way down the neck here, right? So it's thought that the reason young people got hit so hard was that young people often, you don't have a lot of money, right? So if you're going to eat beef, you're going to eat the cheaper types of beef, which are often maybe sausages, and sausages could have who knows what can be mixed up in the sausages, brain, spinal cord, kind of leftovers, right? Um, Whereas, like the nicer cuts of meat, like a steak, again, they can have prions in them, but they aren't going to have such high concentration as like the cheaper cuts, and like the sausages or hot dogs. So that's one reason that it's thought that the young people got hit so hard. It was because of the, the tissues of the infected cattle that they were eating. So then, just to finish the picture, you guys, we think because of feeding the blood and bone meal that had scrapey prions in it to cattle, the cattle developed their BSE prions, and then when we humans ate tissues from the infected cattle, then those cattle, those BSE prions, bound to our good normal cellular prions, causing them to misfold, and that's how we developed our variant Creutzfeldt-Jacobs disease, our ingestive form of CJD. So finally, finally, there was some feed bans. So in 1997, the U.S. Food and Drug Administration (FDA) prohibited feeding most mammalian proteins to ruminants, right? So not using the blood and bone meal, right, to feed to cows, sheep, and goats. And then this kind of troubled me, and I actually don't understand why this wasn't done right from the start. So in 2008, they now prohibit inclusion of brains or spinal cords of animals older than 30 months of age in any animal feed. And I think they mean, I'm guessing, ruminants here. But this is so troubling, you guys, because if you have a cow and it gets slaughtered, right, or made in blood and bone meal, and it's less than 30 months old, that doesn't mean it's not infected, because again, the prions have such a long incubation period. So let's say the calf is infected, early on, right? So they're going to be infected for a long time before they show clinical signs, right? So if it takes 30 months for the clinical signs to appear, that doesn't mean the animal doesn't have prions, right? It's just the prions are multiplying here. We have asymptomatic, right? And then at 30 months, there's enough damage, you have clinical signs and symptoms. So to say that you could eat these animals here or include them in the protein supplements, to me just doesn't make any sense at all. So are there other prion diseases we have here in North America? And the answer is yes. And another really important prion disease of animals that's spreading is called chronic wasting disease. And it's thought this may have evolved here in the United States, possibly in Colorado, right here. So chronic wasting disease is a prion disease of elk and deer. And I've also heard now moose can get infected. And it's thought possibly transmission is similar to scrapie, right? So an ingestive form, right? You can see why it's called chronic wasting disease. So here's a deer that's wasting away, right? And again, all that destruction going on in the brain. They found the CWD, chronic wasting disease prion, in the saliva and blood of infected deer and elk. So we think it started in Colorado. There's gossip that it was a experiment that went wrong, and the experiment, and again, you guys, it's just gossip. The experiment, I heard that their researcher wanted to see if scrapie prions could be transferred from sheep to deer. This is just gossip, you guys, microbial gossip. And so some scrapie infected sheep were penned with deer, right? And apparently the experiment worked, so you had transmission. But then the gossip is some of the infected deer escaped. Now, who knows if that's true or not? Maybe it's just malicious gossip, right? But it seemed to start in Colorado and then it spread north up into Canada, south to the west. I don't think we have chronic wasting disease yet in California, you guys. I hope we'll never get it. And then it spread east. And you might say, well, how did it jump? How did it jump states like this? And again, this is where us humans come in, because there are game ranches where animals, like so-called, well, wild animals like elk and deer and moose would be raised on these game farms, and fed um, feeds containing protein supplements, right, from maybe prion-infected animals, and then people come in and quote unquote hunt the animals, right? So and you guys, I I have I come my family, my mom's side of the family, they were raised in Colorado. Deer and elk was one of the meats they had to eat growing up, right? So I was raised on elk and deer meat too. And so I guess I shouldn't be not respectful of these game farms. But anyway, so why is it that the chronic wasting disease jumped? Because infected animals that had chronic wasting disease would be shipped to other game farms, right? So these animals are being transported by humans. So once again, we see how humans are involved, maybe not only in the emergence of chronic wasting disease, but we're certainly involved in how it's been spread, right? Crazy humans. So the big concern is, can chronic wasting disease, is it zoonotic, number one? Just like in our family, we ate a lot of deer and a lot of elk growing up, right? Could that chronic wasting disease prion jump directly into humans? I would think it's a possibility. And furthermore, in a lot of those areas where the wild deer and elk live, they share pasture or grazing areas with beef cattle. So these look more like dairy animals, but let's pretend these are beef animals. So the concern is, what if, like with scrapie, right, that the chronic wasting disease prions contaminate the environment, maybe the grass, right, and then beef cattle come along and eat the contaminated grass. And now could the chronic wasting disease prion jump into cattle, right? And so that's a huge concern. Never trust a prion, you guys. Never ever trust a prion. So this was 2 years ago, you guys, distribution of chronic wasting disease here. So the red dots are chronic wasting disease in captive facilities. So those are those game farms. So yellow is chronic wasting disease in captive facilities, game farms that have been depopulated. So the same thing like with scrapie, you go in and you just kill everything when one animal's identified. Um, So the dark black is known distribution of chronic wasting disease prior to 2000. So these are on wild, like wild deer and elk and moose. And let's see here, in free-ranging population. So I think what they're showing here with the gray, you guys, is they're showing how it's spread from 2000. So 2000, this is where the wild infected were seen. And so since 2000, it's spread here. All the gray are now the positive area. And again, it's so weird how it jumps, right? But, you know, as humans, hauling our animals around to game farms. Okay, so there's other TSCs of other animals. Folks, some cats, mink can get infected. Squirrels, in folks in some areas of the United States, squirrel brains is supposed to be a delicacy. But you guys, if you can, and I know it can be really tricky, like to refuse food that somebody makes for you, I would so try not to eat brains or spinal cord or tonsils or intestines. I just don't think those are good tissues to eat. And then, in these other slides, you guys, I apologize, these are optional, right? So some people are really fascinated by this, so I was just trying to find some additional articles that might be of interest. So this is just a link to the biological function of cellular prion proteins. This is from a 2017 link. Right, so this is just goes on and on and on, the proposed ideas of their normal function. And then this was some of the newer diagnostic techniques, folks, that they're trying. And some of these, I could not explain these. This is beyond my physics understanding here, but they're trying all kinds of new ways to diagnose prion diseases in humans or animals that are still alive, because we desperately need that diagnostic test. And that's it. Okay, folks, so I'll close this. The next unit we're going to do is on medical microbiology, and we'll start out first with a discussion of virulence factors. You know, what is it that lets a microbe cause disease? And then the second part, we'll talk about the epidemiology, reservoirs and transmission. Okay, folks.